Hello and welcome. My name is Matthew Greer. I am a student of Grand Canyon University. I am going to be presenting today for Bio 515, which is Concepts of Human Physiology. My professor is Dr. John Tassone, and today I'm going to be discussing action potential, why it's important, and how it works, and how we propagate it through our cells. All right, the first question we need to ask and kind of answer is, what is action potential? Action potential are brief, very short-lived, but very large changes in our membrane potential of our cells. And during these action potentials, the potential actually reverses to the inside of the cell becoming positive instead of its normal resting state of being negative. Unlike graded potentials, they are non-decremental. What this means is they don't diminish over time. They can be carried over very long distances. So in this graphic here, like this guy is stubbing his toe, that pain, that new stimulus will travel all the way from his toe through his nerves and his feet, all the way up to his spine, where he will, start to acknowledge this pain inside of his brain. And all of this signal won't be lost. It'll be carried from the toe all the way up to the brain without losing any of that information. Very useful for the body to have. So some important terms that we're going to talk about and that we need to have a basic understanding of before we can really dive into action potential is first equilibrium. You know that you should remember from your high school biology class that objects will move and particles and ions and molecules will move from areas of high concentration to areas of low concentration. So they will try to put as much space between them as possible. So in this passive transport here of diffusion, they're just going to naturally go inside of the cells. Now, not everything can move across the cell membrane. The cell membrane is made of lipids. So things that are charged, things that are big, things that are polar have a hard time moving across this membrane. So we have to use some energy to get them to move across the membrane or to move up their concentration gradients instead of down it. In active transport, we are now using, and this is active transport over here on the right, we're now using energy, this molecule called ATP, to help move things from a high concentration to a low concentration, or from low concentration to high concentration, excuse me. So we're moving things against their concentration gradient. Now, this is done through the use of membrane proteins. And I have this image here that has facilitated diffusion, which is actually passive transport. It's not requiring any energy. But these molecules, for whatever reason, can't naturally pass through or don't pass through fast enough this cell membrane. So we have these proteins embedded inside of the membrane that allow these little green dots to move down their concentration gradient into the cell. And on the other hand, when we're in active transport, usually refer to these as pumps inside of active transport. So they require ATP. It's an active process, so we're pumping instead of just passively letting these substances go into the cell. So another important thing is we're going to talk a lot today about sodium and potassium, these ions that our body uses. They are used to help create this electrical gradient in our cell. A very important protein that you should have seen in your biology class, in your introductory biology class, is the sodium-potassium pump, where we take 3 sodiums and we pump them into the cell, and we remove 2 potassiums out of the cell using ATP. This ATP binds, turns into phosphate and ADP, and then leaves. This act of getting more sodium on the outside, more potassium on the outside, creates an electrical gradient, where the inside of the cell is more negative and the outside of the cell is more positive. So we have this concentration gradient of more potassium on the inside, and we have this voltage gradient of more positives on the outside than on the inside. So this, anytime we have this difference, now we have potential energy that is wanting to kind of come to this balance, wanting to turn into kinetic energy. And inside of the resting neuron, inside of a normal human cell, human body, the resting membrane potential is around negative 70 millivolts. This is an important number. So now that we've done a little review, we can talk about some of these channels, these membrane channels that are very important for action potential and this electrical gradient that we have. The first is going to be these sodium channels. These sodium channels have 3 different orientations they can be in, and the job of the sodium channel is to let sodium in or keep sodium out. And it's got 2 ways of keeping sodium out. It's got this little activation gate in the middle here, and it's got this inactivation gate that kind of dangles down here on the bottom. If either of these are closed, this is unable to allow sodium through. So in this kind of orientation, this is closed, but it's capable of opening. For example, if we move over to this middle one here, it is now open, and the sodium on the outside will be able to flood inside of there. There's a higher amount of sodium in the ECF, the extracellular fluid, than the ICF, the intracellular fluid. So it wants to go down its concentration gradient. It wants to go through this channel. So if this opens, it's going to rapidly go through there. And then once this kind of triggers, this starts to close, this little dangly thing, this inactivation gate. It will close, and it will prevent sodium from coming through here. And then this channel will be allowed to close. This is very important when we talk about refractory period. For our potassium channel, it's a little bit simpler. It has just an activation gate. It will open up and allow this potassium to travel through when a certain voltage is reached. So we need to talk about and define some terms as far as the voltage of the cell, because in an action potential we have this very quick change in membrane potential, and we need to have some terms to describe that. So we discussed earlier our resting potential is negative 70 millivolts down here. We are now resting. If we have a depolarization, or we now have a signal, the cell is going to open up that sodium channel, and we're going to get tons of sodium ions in. This is going to raise our voltage up to around positive 30. When that happens, this is depolarization, very quick process. When that happens, those potassium channels are going to open up, and we are going to repolarize. We're going to get more negative really, really quickly. In fact, it happens so quickly that our cell kind of overshoots and causes this hyperpolarization where it becomes too negative. And then it needs to pump some of those potassium out to get back up to resting potential. Whenever the cell gets to this threshold potential, this very important green dotted line here, that's when we're going to have this depolarization event. If we don't get to this threshold potential, we don't get an action potential. So it's an important that we have enough of a stimulus to set off these cells. But on the same side, it's nice that whenever you walk, it doesn't feel like you're stubbing your toe. We need to have this kind of differentiation between a signal that means something and just everyday signal that our brain kind of desensitizes and filters through. So this kind of graphic ties it all together. So we're depolarizing, this triggering event, stubbing your toe. Once you've reached that threshold, this sodium channel now opens and sodium floods outside of the cell. As this opens up and starts to allow sodium through, the inactivation gate here is the delayed gate, but it is going to start, the minute that this starts to go through, it's going to start that slow process of closing so that we don't get too much of this depolarization. We want to make sure that our cell doesn't go too far depolarized. So it will close and block the influx of sodium there. And as this happens, now we open up those potassium channels, and the potassium floods back out, and we quickly repolarize back down into the negatives here. And then here we're going to have this gate closed again. So we're still not getting any of these, but now we're also not getting any of these potassiums going out either. No sodiums in and no potassiums, and the cell is allowed to go back to a resting potential. So when we say that action potentials are non-decremental and they're really good at long distance signaling, it's important to note a couple things. First, they are all or none signaling. You either have enough of a stimulus to have an action potential, or you don't, and you have no signal going down the neurons. There's no stopping it. When you stub your toe, you don't get to like choose halfway through that signal's not going to your brain. It's going to make it all the way up to the brain. It doesn't stop anywhere along the line. It goes all the way up. we also have this nice refractory period where our neurons need some time to repolarize and get back up to that resting potential. So they can't just depolarize over and over and over again. There's a little bit of time, and it's not long, but a little bit of time between depolarization events. There are two types of neurons in your body. There's unmyelinated, that doesn't have any myelin on it, and then there's myelinated, that has this myelin kind of lipid layer on it. And these guys have different methods of traveling down the neuron. Neuron anatomy has the dendrites, these little hair-like things that receive signals, the cell body that has the nucleus, and then there are long axons that the signal's actually going down. Now, unmyelinated axons, they have the ability to do what we call contiguous conduction. So this conduction is going to run all the way down the whole length of this. It's going to start up here, or wherever the signal was, and it's going to carry on all the way down the axon to the next axon, to the next neuron, to the dendrites of the next neuron, or to the central nervous system, or wherever that information needs to go. The second type of conduction we can have is saltatory conduction, and this is something that we see in our myelinated axons. And what happens is the charge starts, once again, the signal starts up here by the body, and instead of running all the way down, now it jumps in between these lipids. These lipids are insulators, so this part of the cell is not getting, that action potential is not getting excited, but it's jumping to these unmyelinated parts of it. And this is actually a much faster, saltatory conduction is a much faster method of transportation for information. Now, as this charge or this signal from the conduction goes through, these cells that are affected need that time to charge, need that time to get back up to that resting potential for that refractory period. So they're going to relax and then be ready to have another depolarization. And this happens thousands, if not millions of times in your body every day. Every time you have a thought, every time that you want to move your hand, your nervous system is constantly at play here, creating these action potentials, creating these ability for you to interact with your environment and sense your environment. And it's all due to these very awesome cells. These conductions are both fast. Saltatory is much faster because it's jumping. Saltare means to jump, so this signal is jumping from these kind of spots of unmyelinated area there on side of the axon and getting depolarized. So it's a very effective method of taking signals and transporting them around the body. If you would like to do some more reading on this topic, this is where I pulled the information for this presentation. Britannica had some really good graphics on the difference between myelinated and unmyelinated. And then Sherwood, Human Physiology, does a really good job of explaining this concept more. I hope that you enjoyed this topic and that you know a little bit more about action potentials after watching this. Thank you.